Good afternoon and a very warm welcome to all. So today we are going to discuss about the other important pathway in the microbial metabolism of the bacteria. That means Hexose Monophosphate Shunt Pathway. This Hexose Monophosphate Shunt Pathway, it is also known as Hmp Shunt or pentose phosphate pathway. It is also known as phosphogluconate pathway, and this is an alternative pathway to glycolysis and TCA cycle for the oxidation of glucose. So it is nothing but an alternative pathway for the oxidation of glucose. Hmp shunt pathway, it is more anabolic in nature. Anabolic means synthesis of the materials. Catabolic means destruction, or the materials are destroyed or destructed for the releasing energy. In the case of anabolic, it is the synthesis. And this process occurs in cytosol. It is mainly concerned with the biosynthesis of NADPH and pentosis. It is a 5-carbon sugar. Actually, glycolysis involves the oxidation of 6-carbon sugar. In pentose phosphate pathway, it includes the oxidation of 5-carbon sugars. About 10% of glucose entering this pathway per day. So, the glucose 10% are entering this pathway. First, it is the primary route for the formation of pentose. So, the primary route for the formation of pentose is Hmp shunt pathway. The pentose, which is the 5-carbon sugar, and this 5-carbon sugar pentose, it is required for the synthesis of nucleotides. For example, DNA, RNA, etc. and a variety of other essential cellular components. Then, it is also used for the certain redox reactions in the pentose phosphate pathway, which generate NADPH. So, there are certain redox reactions, that means releasing of intermediates, etc. And these reactions which generate NADPH intermediates in the PPP, means pentose phosphate pathway. The glycolysis, TCA cycle and the pentose phosphate pathway, they are known as central carbon metabolism. These three pathways, along with the reaction which convert pyruvate to acetyl-CoA, these three pathways contain all the chemical precursors which are required by the cells for the biosynthesis of nearly all other biomolecules. So, these three are considered as central carbon metabolism. The PPP means pentose phosphate pathway produces pentose phosphates, that is 5-carbon sugar, erythrose phosphate, which is also a 4-carbon sugar, and NADPH. Apart from the 5-carbon sugar, a 4-carbon sugar is also produced, as well as the intermediate NADPH. The pentose phosphates are mainly, I am repeating again the same point. These are required for the nucleotide biosynthesis, while NADPH, what is the function of NADPH? It is main intermediate which is used as a main mobile electron donor for the building reactions or anabolic reactions. This pentose phosphate pathway also produces a 7-carbon sugar. So, I have already discussed it produces 5-carbon sugar, 4-carbon sugar, and also it produces a 7-carbon sugar, which is known as sedoheptulose phosphate, and they are used in the building of gram-negative bacteria's outer cell membranes. So, now coming to the pentose phosphate pathway. The pathway involves several redox reactions and also the multiple molecular rearrangement that interconvert the molecules of 3, 4, 5, 6 and 7 carbons, which results in interconversion of molecules. within the different types of carbons. Now next is the pathway begins with the oxidation of glucose 6-phosphate. And this glucose 6-phosphate, it is a key intermediate of glycolysis. So first of all, in the glycolysis, glucose can be converted to glucose 6-phosphate. Here also, the pathway begins with the intermediate of glycolysis, that is glucose 6-phosphate. With the help of the enzyme glucose 6-phosphate dehydrogenase, that is G6-PDH. This enzyme, what is the function of this enzyme? This enzyme oxidizes G6P, that is glucose 6-phosphate, through the coupling reduction of electron carrier. And this oxidizes means release electrons, loss of electrons. And this may result in the reduction of electron carrier NADP to make NADPH. So this NADP can be converted to make NADPH. So this may result in the synthesis of NADPH. Now coming to there are two different types of enzymes involved in PPP. Transaldolases and transketalases. These are used to produce what? This intermediates within the pathway. Intermediates are NADPH, etc. So these enzymes are involved in the synthesis of intermediates. What will be the net result? The net result will be the oxidation and subsequent decarboxylation. That means removal of carboxyl group of the glucose. So what happens if the 6 carbon from a 6 carbon molecule, 1 carbon is removed through decarboxylation? What happens? It may form a pentose, that means 5 carbon sugar. The total reaction involves 3 glucose 6-phosphate molecules which is being oxidized to form 3 carbon dioxide, 1 glyceraldehyde phosphate and 2 hexose phosphate. You will understand by seeing the diagram. And in this cycle, the formed glyceraldehyde phosphate, they may feed into glycolysis. So, the product which is formed, that is glyceraldehyde 3-phosphate formed during this PPP, this can directly enter to the glycolysis to continue as glycolysis. And also, the 2 hexose phosphates are formed. They can recycle into the PPP or glycolysis. So, the hexose phosphates which are formed in this pentose phosphate pathway again recycled to form the same cycle of hexose phosphate Hmp shunt, or sometimes this may enters to the glycolysis. Hexose phosphate means glucose phosphates, that is glucose 6 phosphates. So this is the reaction. Glucose 6-phosphate molecule. This with the help of the enzyme glucose 6-phosphate dehydrogenase. This is the first step. Converted into 6-phosphogluconolactone. Then it is an anabolic reaction. Why? During the conversion of glucose 6-phosphate to 6-phosphogluconolactone, what happens? Oxidation occurs and this may result in the molecule of NADPH plus H plus. So the building up of the intermediates occurred during this step. So that's why it is called anabolic reaction. Next step is 6-phosphogluconolactone. 6-phosphogluconolactone, which can be converted to 6-phosphogluconate with the help of the enzyme 6-phosphogluconolactonase. This is the enzyme converting 6-phosphogluconolactone to 6-phosphogluconate. And this 6-phosphogluconate, which may converted to what? Ribulose 5-phosphate. Ribulose 5-phosphate, it is an intermediate of pentose sugar. So this with the help of the enzyme 6-phosphogluconolactonase dehydrogenase. Here also again dehydrogenase is coming. This enzyme converts 6-phosphogluconate to ribulose 5-phosphate. Here also what happens? the intermediate synthesis occurs, that is NADPH. That's why anabolic reaction. Okay. Okay. In the next step, this ribulose 5-phosphate, which can be converted to ribulose 5-phosphate, which can be converted to ribose 5-phosphate and cellulose 5-phosphate. So this ribulose biphosphate converted to ribose 5-phosphate and cellulose 5-phosphate. So what happens? How this ribose 5-phosphate is formed? When the ribulose 5-phosphate is acted with the help of the enzyme ribose 5-phosphate isomerase, it may result in the formation of ribose 5-phosphate. What happens here? Here the ribulose 5-phosphate converted to cellulose 5-phosphate with the help of the enzyme epimerase, that is ribulose 5-phosphate 3-epimerase converting into cellulose 5-phosphate. So this ribose 5-phosphate, which may take part in the nucleotide biosynthesis, that is synthesis of DNA, RNA, etc. Okay. The next step is this cellulose 5-phosphate. Here also different enzymes are acting, trans-ketolase and trans-aldolase. We have already said. So the cellulose 5-phosphate can convert it to what? Glyceraldehyde 3-phosphate and sedoheptulose 7-phosphate. So this is a 7-carbon sugar, sedoheptulose 7-phosphate, this one. And this is 3-carbon, glyceraldehyde 3-phosphate. And again the reaction of this to form what? Glyceraldehyde 3-phosphate can be converted to erythrose 4-phosphate or fructose 6-phosphate. So this molecule can be converted to sedoheptulose 7-phosphate and glyceraldehyde 3-phosphate molecule formed from cellulose 5-phosphate, and this sedoheptulose can be converted to erythrose 4-phosphate, which is a 4-carbon sugar, and glyceraldehyde 3-phosphate. Next, this glyceraldehyde 3-phosphate again results in the formation of fructose 6-phosphate. Fructose 6-phosphate is an intermediate in glycolysis. So this fructose 6-phosphate and erythrose 4-phosphate is produced, and this erythrose 4-phosphate with the help of the fructose 6-phosphate again trans-ketolase reaction occur, and this fructose 6-phosphate can be converted to glyceraldehyde 3-phosphate, which may enters the glycolysis process. So that the steps is quite confusing as well as complicated. As shown in the figure above, the products of the pathway include glyceraldehyde 3-phosphate. So we have seen glyceraldehyde 3-phosphate in the pathway reaction. This sugar can be converted or oxidized through glycolysis. So this glyceraldehyde phosphate sometimes they directly enter the glycolysis for further oxidation, or this can be converted to fructose 6-phosphate, can re-enter the glycolysis. So during the procedure of this PPP, fructose 6-phosphate is also formed, and this also can enter the glycolysis, same to glyceraldehyde 3-phosphate. And NADPH, which is a reductant for many biosynthetic reactions, is also made. So the fructose 6-phosphate is synthesized, glyceraldehyde 3-phosphate is synthesized, NADPH is synthesized during the PPP. So this may enter the glycolysis. Which one? Fructose 6-phosphate and glyceraldehyde 3-phosphate. In addition, the pathway provides a variety of intermediate sugar phosphates that the cell requires. So it may provide sugar phosphate intermediates such as pentose phosphates. So I have told that is from ribose 5-phosphate. This intermediates such as pentose phosphates are synthesized for the synthesis of DNA, RNA, and also for the synthesis of some amino acids. And also erythrose 4-phosphate is also synthesized. This can be used for the synthesis of amino acids and 7 carbon sugar